Okay, so even though I left you with some independent work on DNA replication and a diagram to label about the DNA replication fork, I just wanted to create a short video just to sort of bring it all together and make things maybe a little bit clearer for you. So recall at the beginning of the unit, I said that back in grade 11, you guys learned about mitosis in a little bit more detail and meiosis. And if we were to take this unit and fit it somewhere into where you learned about these two things in grade 11, then this helps to sort of put things into perspective. So just prior to mitosis, the nuclear material in the cell is duplicated, including DNA. This process is called DNA replication. DNA replication occurs in all cells before mitosis begins. So it's done right before the process of prophase. And so when we think about, well, how does DNA make a copy of itself? This is the work that I left you guys with. So DNA is a double-stranded material, a double-stranded helix, we call it. And in order to make copies of that letter code that occurs along that DNA strand, we need to split it into two, which is where we then get this replication fork that we have here. So I'm just going to jump down here first. So DNA replication allows both sides of DNA to be copied simultaneously. Each strand of the DNA is used as a template to make that copy because we know there's complementary letters to each letter that exists there. Once the two strands are separated, we now call this the fork. So as this begins to separate and new strands begin to form, that's called the replication fork. And you'll see that there are a lot of different enzymes involved. There's not much I can do to help you remember what the enzymes do in their names. This is unfortunately part of biology where we have a lot of content to take in and a lot of things to just plain out memorize, unfortunately. So if we look at it in a little bit more detail, the actual process itself, once the DNA separates, all of these little proteins will start working at each strand at the same time. And each strand will be copied. So if we could just go through a quick summary, we have one, actually I'll highlight them as we go or underline them. I have one enzyme here, gyrase, will unwind the double helix. Helicase unzips it, so it actually pulls those two strands apart. Then we have something called single-stranded binding proteins that prevent the strands from actually coming back together. There's a better picture of those back here. These little guys here. They'll attach along so that while it's being separated, they don't come back together before that new strand can form. Then we have an RNA primer is formed by something called primase, so that DNA polymerase 3, so DNA polymerase 3 is what initiates at the starting point. So it assembles nucleotides in 3' to 5', but reads the template in 5' to 3'. So if we look at this, just focus on this picture here. A new template strand has to, a new copied strand has to be made in the 5' to 3' direction. So new nucleotides are being added onto this end. If that's the case, and both of your original strands run anti-parallel to each other, this is what lets you end up with one called a leading strand, one called the lagging strand, and I'll explain that in a little bit more detail. So this one, because our 3' end of this template is here, we can actually continuously form or add new nucleotides onto the 3' end in this direction in a continuous motion all the way through, which allows that side to do this more efficiently and more quickly, which is why we call it the leading strand. If we think of the opposite strand, it's anti-parallel, so its 3' end is up here and its 5' end is here. If we're splitting this DNA molecule apart here, in order to create that new strand from 5' to 3', meaning that we're adding on to the 3', then it can only do that in segments as far as it's been unwound. So as the original DNA starts to pull apart and create that replication fork, this one can continuously go as it unwinds. This template or this new strand has to be built from the direction of this direction down. So it's got to keep waiting for more DNA to unwind before it can start making these tiny little segments in the opposite direction, which will eventually be joined by this protein here called DNA ligase. And so because this is created in segments, it's a little bit slower of a process, which is why we call it the lagging strand. leading fast, lagging a little bit slower. And these small fragments have a special name too. They're called Okazaki fragments. Once that whole template has been copied, this DNA ligase will come and join those together to make it one continuous strand like the leading strand. Okay, so again, at the end, I'm just highlighting the important things that you need to know. At the end of this, we have something called DNA ligase. We'll join the fragments of DNA on the Okazaki fragments. So these small little segments that had to be made in sections along that replication fork. And then replication itself will terminate when DNA polymerase reads something called a telomere. And a telomere is just a repeating sequence of DNA that gets removed, and it just signals that DNA polymerase 3 to stop. It's sort of like a stop signal. And then at the end, in order to make sure that things have been replicated properly, there's a proofreading process that happens to the replicated strands that just double checks. This is something that goes along and double checks that the strand was copied properly. And just an extra tidbit of information, in many prokaryotic cells, remember like something like bacteria that doesn't have a membrane-bound nucleus, replication takes under 40 minutes, and thousands of nucleotides are replicated every single second. So think about how fast that process is. And this is why bacteria can make copies of themselves so quickly as well.